السلام عليكم شباب. صباحكم الله بالخير، كل عام وأنتم بخير. Our lecture today has two parts. In the first part, we're gonna talk about the components of the extracellular matrix, while the second part will be about the connective tissue types. The extracellular matrix is the main component of the connective tissue. As we see in the figure, the extracellular matrix is largely composed of different combinations of protein fibers. Surrounding fibers and cells, there are unstained areas which correspond to the ground substance. This ground substance is a complex of proteoglycans, glycosaminoglycans, and multiadhesive glycoproteins. The hydrated nature of ground substance of connective tissue provides a medium for exchange of nutrients and metabolic waste between cells and blood capillaries, the connective tissue. From now then, we will talk about each of the extracellular matrix components one at a time. Now, let's start with the fibrous component of the extracellular matrix. Our connective tissue has three types of fibers. First, we have collagen fibers. These pink structure in this figure. Thinner than the collagen fibers, we have elastic fibers, which have been branched here. Lastly, we have collagen type 3 fibers, which were previously known as reticular fibers or reticulin. Besides, this figure contains blood capillaries and connective tissue cells such as fibroblasts, mesenchymal cells, macrophages, and adipocytes. Let's start with collagen fibers. These red bundle of fibers are the main fiber type found in the most supporting tissue. Collagen is the most abundant protein in the human body, and it has been estimated that it represents 30% of the dry weight of the human body. The black arrows here refer to fibroblasts, which secrete collagen in the form of procollagen molecule into the extracellular matrix. These procollagen molecules assemble together to form collagen fibrils. At least 28 different types of collagen have now been identified in the collagen superfamily. They have been classified according to their morphology, amino acid composition, and physical properties. Collagen can be fibril-forming collagen, network or sheet-forming collagens, or linking or anchoring collagens. Fibril-forming collagens include type 1, 2, 3, and 5 collagen. Collagen type 4 is an example of network or sheet-forming collagen. It is the major structural protein of external laminae and basal laminae of epithelial cells. Linking or anchoring collagen include type 7 collagen. These are short collagen that link fibrillar collagen together to form larger fiber. They also attach the basal laminae to the underlying reticular lamina in the basement membrane. By light microscopy, collagen fibers are acidophilic, thus stain pink with hematoxylin and eosin. Large bundles of collagen fiber are tightly packed and oriented in one direction for maximal tensile strength. In between the bundle of collagen fibers, we have fibroblasts to maintain the collagen fibers. This section is from the dermis of the skin. Unlike the previous section, collagen fibers here run in different directions. They are perpendicular to each other. This is important because they give strength in all directions. This section from adipose tissue with fine collagen fiber coursing between adipocytes and the blood vessels. Here, if you want to look specifically for collagen fiber, you may need to stain it with a special stain. Here, this section was stained with a trichrome stain to demonstrate collagen fiber, and they stain blue with this stain. Here is the typical appearance of type 1 collagen fibrils by electron microscopy. These fibrils were cut transversely, while those were cut longitudinally. A characteristic feature of these fibrils is the presence of cross-banding pattern. This banding pattern results from polymerization of pro-collagen molecules. These molecules are 300 nanometer in length. Each has a head and a tail. There is a gap between one pro-collagen molecule and the next pro-collagen molecule in the same row. The tail of one pro-collagen molecule overlaps the head of the next pro-collagen molecule in the next row. The regular arrangement of pro-collagen molecules, gaps, and overlaps continue as large collagen fibrils are formed. This gives fibrils a characteristic cross striation with alternating dark and light banding seen by electron microscope. Fibrils further assemble to form collagen fibers seen by light microscope. Type 1 fibers often form large aggregates bundled and linked together by other collagens. Now, a question is raised. Why do we have this alternating dark and light bands on collagen fibrils when seen by the electron microscope? Well, heavy metal stains used in electron microscopy deposit on the gap region. Therefore, the dark bands represent the gap region filled with heavy metals, and the light bands represent overlap regions which lack heavy metal deposition. The second type of fibers in our connective tissue is the reticular fibers or type 3 collagen fibers. They stain poorly with hematoxylin and eosin. However, they specifically stain with silver impregnation method. Therefore, they are called argyrophilic. Argyros means silver, يعني هي محبة للسيلفر. They are also stained by periodic acid chief BAS, and both argyrophilia and BAS positivity is due to their high content of sugar. Here we have a representation of reticular fiber in the pancreas. These are pancreatic acini, and these thread-like structure are the reticular fiber, and here the reticular fibers support the pancreatic parenchyma. This tissue is stained with silver, and these black threads are the reticular fiber, and unstained areas represent the pancreatic acini. Here is another example of a tissue contains reticular fibers. This cartoon represents liver sections, and these are the plates of hepatocytes or liver cells. As we see here, these plates are supported by a network of reticular fibers, these black threads surrounding the plates. They are better demonstrated here in silver stained section, and these are the reticular fiber, and unstained area correspond to the hepatocyte plates. Here we have a silver stained section of a lymph node. Reticular fiber appear as delicate network of black fibers surrounding the cells of the lymph node. The black dots here represent the nuclei of these cells. Their cytoplasm is unstained by this method. The third type of fibers in the connective tissue is the elastic fibers. These are thin branching fibers that form networks in the tissues they have. In some places, elastin form discontinuous sheets in the extracellular matrix. Elastic fibers have rubber-like properties. They are stretchable up to 1.5 times of their resting length and recoil to their original shape. Elastic fibers are composed of a core of elastin protein, this green structure, and surrounded by microfibrils composed of fibrillin. Both elastin and fibrillins are produced by fibroblasts. They also can be made by smooth muscle cells, chondrocytes, and chondroblasts of elastic cartilage. During the process of elastic fibers formation, microfibrils made of elastin protein are first laid down. These act as a scaffold upon which elastin protein is deposited. Elastin accumulates and eventually comprises most of the elastic fibers. In the cartoon, we have collagen fibers and elastic fibers, these dark structures. Elastic fibers are thinner than the collagen fibers. To better demonstrate elastic fibers, you may need to stain them with a special stain. Here, there is a section from the dermis of skin stained with a special stain for elastic fibers. These dark structures are elastic fibers and interspersed between the collagen fibers that appear here red. In large arteries, elastin forms discontinuous sheets called elastic laminae rather than elastic fibers. In hematoxylin and eosin stained sections, elastic laminae are eosinophilic. Smooth muscle cells appear in between these elastic laminae. Elastic laminae in the large arteries can stretch, allow these vessels to distend and recoil during the cardiac cycle. This is an electron micrograph of supporting tissue of the mouth trachea. These structures are the elastin molecule deposited by fibroblasts. The elastin molecule are surrounded by microfibrils that cut here transversely, so they appear as dots. In this area, microfibrils are cut longitudinally, surrounding the small areas of elastin. The upper part of the figure show transversely cut collagen fibrils. In the next few slides, I'm gonna talk about the ground substance of connective tissue. The ground substance of the extracellular matrix is a highly hydrated, transparent, complex mixture of macromolecules. It's principally made of 3 macromolecules. These are glycosaminoglycans or GAGs, proteoglycans, and multiadhesive glycoproteins. It fills the spaces between cells and fibers in connective tissue. Because it is viscous, it acts as both a lubricant and a barrier to the penetration of invader. In a hematoxylin and eosin stained section, Ground substance appears as a background of pale blue color between collagen fibers. However, they can be seen more clearly with appropriate special stain, like here. This section is stained by Alcian blue and stains the ground substance blue, which appear between the red stained collagen fibers. Let's start with glycosaminoglycans or GAGs. These are long unbranched polysaccharide chain composed of repeated disaccharide units. These are the GAGs here. As we see, they are often attached to a core protein. This structure is called proteoglycan, and I'm going to talk about it in the next slide, inshallah. GAGs and the proteoglycans carry negative charge, therefore they attract cation like sodium and water. This renders them extremely hydrated, and this allows the passage of water-soluble molecules like nutrient and waste product to pass between cells and capillaries in the connective tissue. And because they are viscous lasciga, they act as a lubricant and impede the passage of macromolecules and bacterial invaders. In hematoxin and eosin stained sections, they are unstained, but they appear as amorphous structure with BS staining. There are 4 groups of GAGs: hyaluronic acid. This is a huge unbranched linear structure. This molecule is unsulfated and does not attach to a core protein. So it is unsulfated or non-sulfated and non-attached to a core protein. The remaining 3 groups of GAGs are chondroitin sulfate and dermatan sulfate, heparin and heparan sulfate, and finally keratan sulfate. Proteoglycans are the second component of ground substance. They are composed of a core protein to which sulfated GAGs are attached. They are synthesized in the rough endoplasmic reticulum and mature in Golgi, where the GAG side chains are added. Then they are secreted from the cells by exocytosis. Perlecan is an example of proteoglycans. The function of proteoglycans include they sequester growth factor like fibroblast growth factors, and this is important in case of wound healing, where fibroblast growth factor is released and stimulate proliferation and maturation of fibroblast to lay down component of the extracellular matrix. In other areas like the filtration barrier of the renal glomeruli, proteoglycans confer unique characteristics on the extracellular matrix, which is selective permeability. There, they impede the passage of large molecular weight plasma protein into the urine. The third component of the ground substance is the multiadhesive glycoproteins. From their names, they act as a glue where they glue structures together. The adhesive glycoproteins are very large molecules with branched oligosaccharide side chains. They provide specific molecular interaction in the matrix by binding cells, matrix molecules, or both. Laminin and fibronectin are the major glycoproteins of the matrix. Laminin, it has a cross shape and composed of 3 chains: alpha, beta, and gamma chains. It is found in the basal lamina, where it's synthesized by adjacent epithelial cells, It can also found in the external lamina surrounding muscle cells and Schwann cells. The arms of the large cross-shaped laminin have binding sites for cell surface receptors like integrin and other components of the basement membrane like heparan, sulfate, type 4 collagen, and intactin. The function of the lamina is to anchor the cell surface to the basal lamina. احنا قلنا ان هو بيبايند السيل سيرفيس ريسبتور انتجرين والكومبوننت بتاع البيسمنت ممبرين عشان كده ان هو بيربط السيلز to their basal lamina. Fibronectin is another glycoprotein in the matrix. Fibronectin binds collagen, heparin, various cell surface receptors, and cell adhesion molecules. Fibronectin plays an important role for cell adhesion and cellular migration through the extracellular matrix. الحقيقة الخلايا بتاخد الفايبرونكتين as a track زي شريط السكة الحديد وبتهاجر على هذا التراك. And this is important during development where cells move from one place to the other, and also it's important during spreading of malignant tumors. The last component of the extracellular matrix is the interstitial fluid, and this will be discussed in the next slide. Water in the ground substance is referred to as interstitial fluid. It has a unique composition of the blood plasma. Interstitial fluid is important for exchange of nutrient, oxygen, waste products between cells and capillaries. There are two main forces act on the water in the capillary. The hydrostatic pressure. This is formed by the pumping action of the heart, and this forces water out of the capillaries. The colloid osmotic pressure. This favor drawing water back to the capillary. الاسموتيك بريشر بيسحب الووتر للكابيلاري، لكن الهيدروستاتيك بريشر بيطلع الميه من الكابيلارز. At the arterial end, the hydrostatic pressure is much higher than the colloid osmotic pressure. فالنتيجة إن هنا من الميه بتطلع into the extracellular matrix. على النقيض at the venous end, osmotic colloid pressure much higher than the hydrostatic pressure. فالنت results إن المية بتسحب باك للكابيلاريز. الحقيقة مش كل الانترستيشال فلويد بيروح باك للكابيلاريز. Instead, excess tissue fluid is taken by the lymphatic capillary. We have finished the first part of today's lecture. Now, I'm going to talk about the classification of connective tissues. Connective tissues are classified based on densities of cells, fibers, or extracellular matrix components. They are classified into connective tissue proper, embryonic connective tissue, and specialized connective tissue. The connective tissue proper is classified according to the predominance of cells or fibers into loose connective tissue or dense connective tissue. The dense connective tissue is further classified according to the predominance fiber type into dense collagenous connective tissue or dense elastic connective tissue. Let's start with loose connective tissue. In this tissue, there are numerous connective tissue cells shown among connective tissue fibers. Here we have fibroblast, plasma cells, leukocytes, macrophages, and adipocytes. This type of connective tissue fills in spaces such as lamina propria underneath the epithelium, for example, the epithelium of gastrointestinal tract. Also, it can be found in the dermis under the epidermis of the skin. It is well vascularized, so it's commonly found underneath the avascular epithelium. Because its low fibrous content, it is not very resistant to stress. Loose connective tissue is characterized by loosely arranged connective tissue fibers, abundant ground substance, tissue fluid, and a lot more connective tissue cells. These empty spaces correspond to the ground substance which are extracted during preparation of this section. Here we have thick wavy fibers, ribbon-like. These are collagen fibers, while the thin straight branching fibers are elastic fibers. In this section is another place where you can find the loose connective tissue. Is the lamina propria of the GI tract. Here is the epithelium lining the gastrointestinal tract. Because this is a site of microbial invasion, we see here a lot of immune cells. In this enlarged part, we have plasma cell, eosinophil, and macrophages. Dense connective tissue has fewer cells and a clear predominance of fibers over the ground substance. Dense connective tissue is less flexible and far more resistant to stress than loose connective tissue. It is classified according to the predominant fiber into two types: dense collagenous or white connective tissue and dense elastic connective tissue. These further classified according to the orientation of their fibers into regular or irregular. Dense irregular white fibrous connective tissue is characterized by few connective tissue cells and a lot more connective tissue fibers. Here it has a lot of collagen type 1 fibers interlaced with elastic fibers and reticular fibers. Reticular fibers not seen here. As we see here, the collagen fiber run in different orientation. This provides resistance to stress from all directions. Fibroblasts are visible among the collagen fibers. The dermis of the skin and the capsule of organs are typical examples of dense, irregular, white fibrous connective tissue. This section was taken from the mammary gland. Here we have loose connective tissue surrounding the glandular epithelium and dense collagenous connective tissue for comparison. As we see here in this enlarged part of the loose connective tissue, it has a lot more connective tissue cells with fewer fibers. On the contrary, the dense irregular white fibrous connective tissue contains bundles of collagen fibers run in different directions with few connective tissue cells in between the bundles. Here's a photo micrograph of the dermis of the skin, which is a good example of the dense irregular collagenous connective tissue. Thick, coarse bundles of collagen fibers run in different orientation. In this enlarged part, few connective tissue cells, probably fibroblasts, are scattered among the bundles of collagen fibers. Another type of dense white fibrous connective tissue is the dense irregular white fibrous connective tissue. Thick bundles of collagen fibers are tightly packed and run parallel to each other. In between the bundles, we can see fibroblasts. These empty spaces represent the ground substance. Structures formed by this arrangement are strong and resistant to stress. A typical example of dense regular white fibrous connective tissue is the tendon, and here, as we see, parallel bundles of collagen fibers. And in this enlarged bar, we can clearly see the nuclei of fibroblast between collagen bundles. Unlike dense collagenous connective tissue, elastic fibers predominate in the dense elastic connective tissue. Here is a longitudinal section of dense regular elastic connective tissue, where the elastic fibers are arranged in parallel array. Therefore, it is described as dense regular elastic connective tissue. In large arteries, elastin forms discontinuous sheets called elastic laminae rather than elastic fibers. Collagen reticular fibers and ground substance are interspersed between the elastic laminae. Smooth muscle cells are also found in between the elastic laminae. Elastic laminae are eosinophilic in section stained with hematoxylin and eusin. These laminae are irregularly arranged, and hence this tissue is called dense irregular elastic connective tissue. Smooth muscle cell nuclei are found in between the elastic laminae. Elastic laminae in large arteries are able to stretch, allowing this vessel to distend and recoil during the cardiac cycle. Reticular connective tissue is a specialized type of connective tissue. In this tissue, type 3 collagen fibers form a delicate network to support various cell types. Reticular fibers are produced by a specialized type of fibroblast called reticular cells. Reticular cells remain associated with and partially cover reticular fibers. In this silver-stained section, as we see here, these black threads are reticular fiber network to support the parenchyma of the hematopoietic tissue bone marrow. This cartoon represents the pancreatic acini and the reticular fibers surrounding them. In this silver-stained section, reticular fibers are black, small, and don't form bundles. They are arranged in the form network to support the pancreatic parenchyma. This cartoon represents a mesh-like network of reticular fibers that support liver cells and hold them together. In this silver-stained section, reticular fibers appear black and they support the parenchyma of the liver. The cytoplasm of hepatocyte is unstained in this preparation. There are empty spaces here between the reticular fibers, and these are the liver sinusoids.